Thank you so much, Perry and Murray, for those very kind introductions. So I'm speaking to you today from Garramuggle land, also known as Manly, and I pay my respects to the Garramuggle elders past and present. I want to thank the organising committee, especially Katya, Perry, Alana, all the folks doing such a great job for this fantastic meeting. I've seen already so many great talks, especially from the next generation, so it's been great. Thank you again. It's a huge honour to receive the medal, and I'm again grateful to the Society. Ian Meiosis was a really incredible scientist, and by all reports, as Murray said, a thoroughly decent human being, just like so many entomologists I've ever met over my career. And Ian actually did a little bit of work on cockroaches, but his partner, Jo Meiosis, was an absolute pioneer in Australian cockroach systematics. And I've been very fortunate to be able to have the opportunity to build on her work. So you can see Ian and Jo there. All right. Now, can I move this forward? Oops. There we go. Okay, so this is an artist's depiction of a landscape from the Carboniferous period. About 300 million years ago it ended. And although we can't see them, this landscape would have been absolutely teeming with cockroaches. And we know that because there's a lot of Carboniferous fossils of, well, cockroach-like insects. They're not like modern cockroaches, they're slightly different, but we call them rochoids. And basically, these insects probably had a pretty similar lifestyle and had probably similar diet to the cockroaches of today. And this little video, I hope you can see in the bottom right, is a video from Mark Sutton doing some X-ray tomography, and he's shown that the mandibles and the wing venation in that particular 300 million year old fossil is very similar to the cockroaches of today. Okay, so if we fast forward 300 million years ahead, the descendants of those rochoid insects, which are the termites, some of the descendants of the termites, and they are now dominating a number of subtropical and tropical ecosystems throughout the world. And they have incredibly huge biomass, really up there in the top 10 of all kinds of animals. And here we see some of the amazing fairy circles on the Namib plains, and it's fairly certain that these are being caused by termites, although it's a little bit controversial. And over here we see a bunch of Cryptocircus mounds from the Campos Cerrados in Brazil. And of course, we have an incredible amount of termites in Australia, and they are very strong in the top end. So we see some magnetic termites down here, and we also have the tallest termite mounds in the world. They get up to about 7 meters in these cathedral termites here. And so given that the termite's only about 3 millimeters tall, these mounds are about the equivalent of, in human terms, of about 4 Burj Khalifas stacked on each other. So termites are pretty impressive when it comes to architecture. Okay, so today I'm going to talk to you about various bits of work we've been doing in the lab over the last several years. The first thing I'm going to talk about is, the talk's in 3 parts, and I'm going to start off with some work on the evolution of symbiosis in termites. And then the second part, we'll have a little bit of a look at the historical biogeography of cockroaches and termites. And then the last part, the most technical part of the talk, will be this story about asexuality in these Japanese dry wood termites, Glyptotermes nakajimai. We'll look at some inbreeding and weird chromosome chains in those termites, and how population genetics has revealed hybrid asexuality. Okay, so this is a phylogeny of cockroaches and termites, and it's based on the mitochondrial genome. And it was mainly done by Thomas Bourguignon, shown here. And he started that work in Theo Evans' lab as a postdoc, and he came to my lab, and we kept working on it. And basically, you got the time scale of evolution down here, going from about 400 million years onwards. And these numbers here are the fossils that we use to calibrate the rate of evolution in order to determine all of these divergence times. And we see some familiar faces over here. This is Periplanet americana, which happens to be reasonably close related to Trionicus from Australia, the termites, Cryptocircus, and Anaplectids. And here's another familiar face here, Blatella germanica. It happens to be related to a number of ectobids. It's an ectobid itself. It's related to this tiny little Adaphylla, which is only a couple of millimeters long. And it's also related to the heaviest cockroach in the world, of course, the soil burrowing cockroaches that Perry's done a lot of nice work on. Now, one interesting thing about cockroaches and termites is that they picked up protozoa. The ancestor of termites and Cryptocircus picked up these protozoa you found pretty much nowhere else from the Parabasilid and Oxymonad phyla. And basically, these protozoa are really densely packed in the gut. So you might not be able to see it too well, but this is a Mastotermes gut that I've pulled out and took a video of. And you can see the Mixotricha protozoa really moving around frantically in there. They basically move all night and day. And this is a bit more of a close-up of a different species, but nevertheless you can see how well packed they are. And so if you think the traffic is bad in your local area, try living inside a termite gut. It's really crazily packed with microbes, and these microbes can sometimes reach about 2/3 of the total weight of some termite species. Now, these protozoa, they're basically found in all termites except for one family, the termitid family. But all termites, and in fact all cockroaches, have a very impressive and complex microbiome of bacteria and archaea, mainly bacteria. And in termites you can find up to about 550 different bacterial genera and up to about 5,500 species just in the one worker termite. So the protozoa, interestingly, have their own symbionts. So we heard a really nice introduction, an overview of Intracellular Symbiosis from Marcus on Monday. And these are the kinds of bacteria he was talking about. These ones are living inside the protozoa, and they actually fix nitrogen from the air. Air's able to get in through the termite gut wall, and those protozoa need that nitrogen, which gets turned into amino acids and protein, because they're feeding on pure wood fragments, which has pretty much no nitrogen. And interestingly, the protozoa also have bacteria attached to their walls, these ectosymbionts that help them with locomotion, and they help them swim around basically in the termite gut, which is an advantage, as you just saw. And so this is a video taken by my collaborator Yuichi Hongo, and you can see all the movement. I hope you can see through the video there, all the movement on the outer wall of the protozoan, and that's basically thousands of these ectosymbiotic bacteria that are flagellating and moving around. And you can see there's another kind of species here, this larger one. There are a couple of hundred of those also found on the outer layer of the protozoan. So the protozoan themselves are full of bacteria. And a few years ago, Yuichi and I were interested in seeing in more detail how many bacteria can be on these protozoa. So these are symbionts of symbionts, and we're wondering, is it possible that these symbionts have their own symbionts? And indeed they do. So this photo here is showing in green the ectosymbionts that are attached to the protozoan. You can't really see the protozoan very well, but we basically design a probe that attaches to the DNA of the ectosymbiont specifically, and then this red is showing a symbiont that's attached to the symbiont of the protozoan. So a bit of a close-up here. You can see that they're quite intimately associated with these. The red ones are quite intimately associated with the ectosymbionts that are joined to the protozoan. And so this is work done by Utami-san in Yuichi's lab. And we also were able to sequence the genomes of the bacteria, the symbionts that are associated with these protozoa. We basically were able to separate the protozoa and then sequence the DNA and then piece back together the genomes. And what we see in this little diagram here is, here we've got the protozoa here and the ectosymbiotic spirochete attached to it. And then this little red bacterium here, which we named Termite tenax, it basically feeds on little cellulose fragments and converts cellular bios into glucose and then into acetate. And that acetate can be taken up by the termite and then used for energy by generation of ATP in the citric acid cycle. And then this little guy, Termite tenax, produces hydrogen, which is then used by the ectosymbiotic spirochete, which turns it into more acetate. So there's lots of complex metabolic reactions going on. This is just one of many. Okay, so we were interested in understanding how did termites get so many bacteria, like up to 5,000 species. How have they evolved such a complex microbiome? And so this is more work we started with Thomas and Theo as well. And so around the time we're starting, Phil Hugenholtz's team had published this paper, and they concluded that vertical inheritance is the primary force shaping termite gut microbiomes. So they were basically using, basically they sequenced the microbiome of the individual termites and then used different kinds of metrics to determine this result. And soon after this, a different viewpoint came out from Andreas Brun's group, and they said that diet is the primary determinant of bacterial community structure in the guts of termites. So here we can see that each of these points represents the community of the different bacteria in a termite gut, and humus feeders tend to cluster together, as fungus feeders tend to cluster together, wood feeders tend to cluster together. So we wanted to take a slightly different approach to understand this problem, because, firstly, a couple of problems with these, not problems, but, what's the right word? Yeah, they could have been better. There weren't that many termites used in either of these studies that I'm showing you here. So we decided to get a decent number. We got 94 different species from across the termite tree, and we took the guts out, extracted the DNA, and then we did metabarcoding on the 16S ribosomal RNA gene of the bacteria. And that basically enables you to tell every last little bacterium, in theory, that is in the termite gut. Yeah, and so we got those all sequenced using next generation sequences. Then we put them all into a big pool. Okay, we combined them, and then we basically sorted them by sequence similarity. And so in our case, like this approach that we used was basically looking at the phylogeny of every last little species rather than combining them into a community before analysis, like the previous studies that I mentioned did. And another feature of our study was that we compared all of the termite gut bacteria with everything else out there on GenBank. So we found all of their relatives, every last thing that we could find from soil, from other animals, from waste processing units, these sorts of things. And we got them all, and then we basically did a large phylogenetic analysis of all those different groups of bacteria that we could find. Okay, so this is a bit of a busy slide, but it's basically showing about 6 of the trees that we got out of a total of 211. So we focused on 211 major groups, which comprised the species that had kind of fairly high abundance in the termite guts that we looked at. And what you can see down in the bottom here, bottom right-hand corner, is the phylogeny of the termites themselves in different colors. Okay, and over in the trees, those colors basically represent the termites. And every time you see an asterisk, that means that it's from it's non-termite. So it's from the environment, another animal, the soil, etc. So interestingly, what we found was the majority of these 211 trees of the termite guts, 130 of them, basically had huge clusters of termite-only sequences. Okay, and a relatively small number had mainly termite sequences, but a few environmental sequences, and then the remainder were kind of a mixture of the termite sequences and environmental sequences. So these seem to be pretty much environmental bacteria that are going through the termite gut. Okay, so what we conclude then is that most termite gut bacterial species, more than 50%, are found basically only in termites. And we're pretty sure, because there's a lot of sequences in GenBank from all sorts of environments, we never, when we do the phylogenetic analyses, all of those termite gut bacteria always cluster together. So this suggests what's happening is that they are occasionally transferred between termites over evolutionary time. So they're kind of adapted to the termite gut, and they get moved across now and then. And so it's basically a combination of the vertical transfer from the colony to the offspring, and horizontal transfer, or what we call mixed mode. It's a term coined by someone else that's occurring in these bacteria. Yeah, and so, of course, many other bacteria in the termite gut are less specialized, and they're found in the guts of other animals or the soil, and they seem to be passing through. And it's a bit hard to explain, how these termite gut bacteria get moved between different termites, but one possibility is that they're transferred when termites interact in the field, and termites often compete over resources, over food resources, and they often fight and they eat each other. So one possibility is that they are basically being transferred during fights. And I'll show you a few of these fights. So these are videos from my collaborator Jan Shabotnik, and you can see they're very, very nasty. Some of these termite soldiers can really inflict a lot of wounds on their competitors. And often we think after this happens, after there's a fight, the winner will often not cannibalize, but eat the others, eat the wounded to get some extra protein. And during that process, perhaps these termite gut bacteria are being transferred. So here's another one. This is the so-called bite and snap. Apologies to those who don't like to see violence on the screen, but it's a very nasty mechanism there, decapitating that little worker there. But some termites have actually lost their soldiers, and they've only got workers, and the workers have come up with a very interesting little way of defending themselves. They basically defecate on their predator or their enemy, and so again, this could you can imagine lead to some possible transfer of these bacteria that we've been discussing. Okay, so let's move on to something completely different now. We're going to look at the historical biogeography of cockroaches and termites, something we've been interested in for quite a while. This is work that I've done a lot of work with Simon Ho, who's the master of molecular clocks. And so cockroaches, they're around about 4,500 species. They're not a particularly large order, but there are many beautiful ones, especially in Australia. We've got some of the best looking cockroaches in the world. And then, of course, there are about another 3,000 described termite species. So the whole group is probably around about 8, 9, 10,000 species once you count all the undescribed ones. Okay, so this tree is derived from the one I showed you at the beginning. It's again from Thomas's mitogenome analysis of, I think we did about 130 different cockroaches from across the 9 or so families. And in this particular version of the tree, what we did was we just took every genus that we'd studied, and then we looked at the distribution of those genera, and we labeled those here in different colors. And those colors, like neotropical in this case, refer to the colors that you see over here. And so this is showing the breakup of the continents over time from about 160 million years onwards. And in this tree, we've got millions of years ago down here. So the ancestor of cockroaches branched off probably from the ancestor of mantids about think it's about 250 million years ago, and then the last ancestor of cockroaches was existing perhaps 220 million years ago. And these numbers here are showing you examples of the influence of tectonic movement on the distributions of these cockroach genera. So tectonic movement being shown here. Now, this is obviously a somewhat preliminary. We haven't got anywhere near the full number of cockroach genera that are out there, but it's a start, and we can make some interesting observations with this particular tree. So, for example, here we see, at this particular branch here, we've got Guyana, which is found in Africa, and a number of Blabrera genera, including Blaberus, Blaptica, Eublaberus. They're all found in South America. Okay, so neotropical. And interestingly, so this particular branch here happened to occur at about 100 million years ago, and that seems to be about the time that these two continents were separated. The separation started around 130, but was apparently complete by about 100 million years ago. So that suggests that this divergence was caused by tectonic movement and vicariance. So another example is down here. So South America was thought to be basically in contact with Antarctica and Australia through this route here up until about 100 million years ago. And what we see here is this group here, Carbonaria and Babiancoa, they're found in Australia only, and this group diverged from Ischnoptera, which is only found in the neotropics, in South America. And again, that divergence seemed to occur about 100 million years ago, about the time you would expect, given that South America was then separate from this land mass. All right, another example is down here. So here we've got, again, these neotropical genera found in South America, Euphylodromia and Amazonina. They seem to have diverged about 150 million years ago, in this case, from a number of taxa that are found in Australia and Indo-Malaya. And so, again, it's somewhat congruent with the idea that this movement did really break up these, or cause the speciation back at this time point. Okay, and then one more. We've got here Diploptera, which is found in Australia and Indo-Malaya. And then we've got an African group and a Madagascan group, which I'll come back to. But basically, this divergence here around 125 million years ago is consistent with the idea that Africa became separate from a number of other continents around about this time, around about 100 million years ago. Okay, so of course there are lots of cases where it seems that dispersal or oversea dispersal must have occurred, and one example comes from this same clade, because we've got these Madagascan genera only found on Madagascar, and we know that Madagascar was separated from Africa probably by about 100 million years ago, if not earlier. And however, this divergence here between Rhiparobia and these Madagascan genera occurred about 50 to 70 million years ago. So, or more like about 70 million years ago. So it's probably more likely that was dispersal rather than tectonic movement. Okay, now this is another example of we're pretty sure dispersal must have occurred. This genus Bolta is found in many different spots. It's found in the Palearctic, Africa, Indo-Malaya and Australia, and yet it diverged from its sister group Ellipsidion, which you might know of. It's a very cute little cockroach, about 70 million years ago, and we know that all these continents had already separated well before about 70 million years ago. Okay, now we'll just take a bit of a close-up look at a couple of genera briefly, and the first one is Cryptocircus. So Cryptocircus, this work was done with Shirley Kerr, who came over for a sabbatical, and this is her with her husband, who's also a cockroach systematist. He came over briefly as well. And Cryptocircus has a fairly interesting distribution. It's found in the Appalachians in North America, as well as some of the redwood forests up in Northern California, Oregon. And it's also found in southern China as well, and moves up. It's found in a few spots up in Korea and also eastern Russia and northeastern China. And this shows some of the habitats that it's found in. So this is a northwestern American forest, Appalachians, Changbai, which is up in near Korea, and the Hengduan Mountains, which are down here. And so Shirley did again a mitochondrial genome study of a bunch of cockroaches from China, Korea, and eastern Russia, as well as North America. And what we found was basically that the Cryptocircus ancestor appears to have lived around about 70, 80 million years ago and diverged into the North American group up here. And the two spots in North America, west and east, seem to have diverged around 35 million years ago. And the Asian ones seem to have diverged also from around about 30 million years ago. And the earliest branches are in the northern groups, the northeastern groups here, as shown here. And these results are pretty consistent with the idea that there was a very large boreotropical forest up near the North Pole about 60, 70, 80 million years ago. And what seems to have happened is that it got a bit too warm from that point on towards the Oligocene, and that warmth would have not suited Cryptocircus because they prefer much more cool mesic environments, and so it seems that they were forced into the remaining refugia that had those fairly high altitudes as time went on. Okay, so we're now going to talk about Nasutotermes, and this is a genus that most entomologists will be familiar with. They build a lot of mounds around the place down southern Australia, northern Australia, and they also build a number of arboreal mounds. And we were interested in, when they came to Australia, how long have they've been building these amazing cathedral mounds? Where did they come from? These sorts of questions. And this work was done by my PhD student, Dej Arab, who just graduated yesterday. And basically, Dej sequenced a number of mitochondrial and nuclear genes, and we got a phylogeny of Nasutotermes as well as Tumulatermes, but I'll just focus on the Nasutotermes for now. And we were able to get a number of related taxa from GenBank, from other people who'd worked on taxa all over the world, South America, Asia, Africa. And what we basically found, briefly, so I can make sure I finish on time, is that it looks like all of our mound builders in Australia have evolved from arboreal nesters. And so basically in green here, we've got all of the arboreal nesters. We've got some arboreal nesters from Asia here. We've got a couple of Australian ones here, and a couple from what's that one? Asia and Africa down here. So what we think has happened is that the Australian lineages have arrived in Australia from the location's not clear, but perhaps Asia or Africa, again through overseas dispersal, rafting of some form during catastrophic weather events perhaps. And what we see is that actually I said they were Australian, they're actually South American, sorry. So those ones are South American. And the major group of Nasuts in Australia, shown here, seems to have arrived about 10 million years ago. And Trioideae is the one that builds the very large mounds. Magnus also builds very large mounds. It seems that these have evolved from ancestral arboreal nesters. So Walkeri you see around the east coast of Australia. Exidiosis goes all the way in the southern part of the continent. And so, yes, we think that mound builders basically they're not that old. They've arisen in the last 10 million years. And this is work we did with a number of people. Again, Theo, Dave Yates helped us out with this one too, and Steve Cameron, so had a lot of help along the way. All right, so just to summarize that little section, so reconstruction of cockroach ancestral ranges suggests plate tectonic movement has had strong influence on their distributions. Dispersal over water also appears to have occurred on numerous occasions. The Cryptocircus ancestors evolved about 80 million years ago in the Northern Hemisphere and retreated to cool mesic areas in mountainous ranges. And the Pseudotermes mound builders evolved from arboreal nesters, which arrived about 10 million years ago. Okay, the last little section here is the evolution of this interesting little termite, Glyptotermes nakajimai. Okay, so as we all hear all the time, sexual reproduction is the norm. It helps to, mix genes so that organisms can adapt to complex and changing environments. Nevertheless, you do see a number of asexual lineages out there of all sorts of kinds of animals and insects, and these are some examples, all from Hymenoptera. But basically, it's thought that these asexual lineages, it's easier for them to evolve when the male isn't doing much. So that if the male's only contributing gametes and not doing much else, then it's easier for asexuality to evolve, and it can give the lineage a much greater reproductive advantage, a twofold reproductive advantage. So this is a good example. So in Hymenopterum social insects, the males are pretty much useless. They're just produced once a year or a couple of times a year, only for reproduction, and they die and they leave their sisters and mothers to do all the hard work in the colony. Now, termites are different. Termites always have males, except for the case I'm about to tell you, and the males typically comprise about half the colony. And so this slide here shows a young termite colony, and we see the king and the queen here, and dad is always helping out. He's always feeding his kids, grooming them. And we see a few both male and female soldiers ready to defend mum and dad and the rest of the colony. So it's more of an egalitarian system in termites. So males are basically seem to be playing an important role, because basically every last termite species you look at, males are there, approximately in half, 50% of the colony. So there's another subtle way, though, that males might be benefiting termite colonies, and that's because termite colonies, a lot of them are basically found in very large logs, which basically very large amount of food and good security. So the nest represents a very long-term useful resource for termites. And what that means is that often when the king dies or the queen dies, he and or she will be replaced by a worker. Okay, that'll then turn into a reproductive. They're suppressed until the king or the queen has died, but what that means is that when the workers evolve into or develop into reproductives, that there'll be a lot of inbreeding going on, and that's a fairly common process in termite colonies. But that's a problem, obviously, because it can lead to the potential expression of deleterious alleles. Yeah, but termites may have come up with a potential solution to this issue, and basically it involves these very weird chromosome systems. And Peter Lokes in the 70s discovered that termites can have these very strange rings or chains when you look at the male meiotic chromosomes at metaphase. So just to remind you how meiosis works, so the bivalents line up, there's recombination, and then they basically get separated by random into two different cells, which then will separate into the gametes. And basically, Soren and Lokes, in this classic study back in the 70s, hypothesized that this ring structure is basically allowing a number of the chromosomes to segregate together. So there's there's basically strong linkage between the chromosomes, so that they're all basically like Y chromosomes. And so you can see here with this structure, the idea is that these chromosomes will always be inherited together. These Y chromosomes will always be inherited, and the neo-X chromosomes will always also be inherited. But in the females, the X chromosomes can actually go under a combination. And so the upshot of this system, I hope I've explained it reasonably well, is that males should be more heterozygous in theory, because they've got a whole bunch of Y chromosomes that never recombine. Okay, so just to recap on males and why they're important. They're ubiquitous, they help in the colony, they contribute phenotypic diversity, because often they're a bit smaller than the females, so that may be beneficial for task specialization. And they also contribute genetic diversity. Plus, they've got these meiotic chromosome chains that may help to sustain heterozygosity under inbreeding. Okay, got to move pretty quickly. So basically, can all female termites exist? The answer is yes. So Toshi discovered these a couple of years ago, and just after that, he won JSPS traveling postdoctoral fellowship and came and worked in our lab for a couple of years. But just before he came, he did a lot of sampling, and he did a few experiments on live termites, and he brought all the dead ones out to Australia, and we did various experiments on them, morphological, chromosomal, and molecular. And so Toshi was basically able to show very clearly that these colonies are definitely all female. The queens never have any sperm in their spermatheca, whereas I should say that there are also sexual populations of this species. So there's asexual lineage, and then there's a sexual lineage. The sexual lineage, the females always have, the queens always have sperm in them. Okay, and interestingly, in the asexual populations, the all-female colonies, yes, the eggs can hatch, as you would expect. But interestingly, a couple of eggs can also hatch in the sexual females. Not that many, but a small number, even though they're unfertilized. Fertilized eggs, very high hatching success. Okay, and so this is basically showing you a map of where the sexual populations are in Japan in the squares here. There are 4 different sexual populations, and the circles are the asexual populations, mainly here. And we did some mitochondrial work and showed that the asexual lineage, all the individuals seem to be very, very closely related to each other, and the sexual lineage, they're all closely related to each other, suggesting that it evolved once, but that's not the case, as I'll show you. So we then went and did some DART-seq analyses, and what you can see is that the sexual populations here, all these in squares, they all cluster with each other and then quite closely to the other sexual populations. But there are 2 very different asexual lineages here. Okay, and then we did a structure analysis, and it suggested that the best number of populations to explain the number of data is about 10, shown here. And we also looked at heterozygosity, and we found that as predicted from, if these males have the chains that I mentioned before, they should have high heterozygosity, which they have significantly higher, maybe 20 to 50% higher than females of the sexual lineages, SL there. But the asexual lineages have really, really high heterozygosity. This is based on about 4,000 SNPs. Okay, and lo and behold, when we looked at the male meiotic chromosomes, they have a very large number of chromosomes that are linked. So they form about chain of about 30. So what that means is about 15 of the 17 chromosomes they have get inherited as a unit. But standard male mitotic is 2n equals 34, and same with the sexual females have 2n equals 34, but the asexuals have 2n equals 35. So I mistimed this, but what we have concluded is that the asexual lineages arose from 2 different hybridization events, okay, between a 2n equals 34 and a 2n equals 36, These two we can't see anymore, but this is the best way to explain how these asexual lineages have arisen. Yeah, so just to summarize, we think that this can happen because, I mean, the asexual lineages have evolved because unfertilized eggs of sexual lineages can hatch, as I showed you before in here. Sexual colonies usually have multiple queens, and that's required for any kind of incipient termite colony because they need to groom each other. And the hybrid asexuals have much higher heterozygosity than sexuals, and this potentially provides a fitness advantage and may explain why they've taken over. It appears they've taken over some of those populations. Okay, so this is the only known termite with all female colonies, and males in sexual lineages have among the highest number of known meiotic chromosome chains, 15 out of 17 of any organism, allowing enhanced heterozygosity. And the evolution of asexuality in G. nakajimai is novel because males appear to provide benefits in the sexual ancestors other than gametes, but nevertheless, they've been taken over by the asexual lineage. Thank you so much for listening. I've got a lot of really great collaborators, all these folks here. And this is a photo of my lab. We've got Toshi there, Simon here, Dej. This is a photo of Perry a few years ago. And thanks to all these people for being such great collaborators. I will stop there. Thank you.
Great. Thank you so much, Nate. That was a fantastic talk. I'm probably a little bit biased, but I'm sure everybody else thought the same. So I think we've got maybe time for one question, if anybody would like to put that in the chat. Otherwise, you're more than welcome to contact Nate afterwards, and please ask him as much as you like.
All right, hang on a second. All right, I got one. Can the switch from arboreal nests to mound building in termites be linked to increasing temperature and aridity? Yes, that seems to be the case. So, you know, everyone knows a story about the increasing aridity over the last 10 million years. And presumably those arboreal nesters kind of 10 million years ago, they might have got in and moved west. But when things started drying up, being closer to the ground probably would have been a better option because maybe it was too hot and dry up in the trees.
Okay, great. So Nate, if you want to contact any of the other people who have asked questions through a direct message or something like that, feel free to.